Welcome back to the Muse B Research Cell Culture series. Today we're covering one of the most critical skills in any cell culture laboratory, aseptic technique and preventing contamination. You can design the perfect experiment. You can select the right cells. You can plan your controls carefully. But if your cells become contaminated, your data is unusable. Sterile technique is not optional. It is foundational. Aseptic technique refers to a set of practices used to prevent contamination by unwanted microorganisms such as bacteria, fungi, yeast, mycoplasma. In cell culture, we are creating a highly controlled artificial environment. Our mammalian cells grow slowly. Bacteria grow very fast. If contamination occurs, microbes can outcompete your cells, change media pH, alter gene expression, produce toxins, destroy months of work. The goal of aseptic technique is simple: protect the culture from the environment, and the environment from the culture. The most important tool for sterile work is the Biosafety cabinet, also called a laminar flow hood. It provides HEPA-filtered air, laminar airflow, protection for the sample, protection for the user. Quote, laminar airflow means air moves in a smooth, uniform direction. This airflow prevents contaminated room air from entering your workspace. Important reminder: the hood does not make you sterile. Your technique does. Poor habits inside a perfect hood still cause contamination. Preparation begins before you even touch your cells. Start with basic lab hygiene. Tie back your hair, remove watches and loose jewelry, and wear a lab coat and gloves. Before working, disinfect your gloves with 70% ethanol. Wipe down all bottles, media, and flasks before placing them inside the hood. And only bring in the materials you actually need. Clutter inside the hood disrupts airflow and increases the risk of contamination. Once you're inside the hood, technique becomes critical. First, work from clean to dirty. Keep sterile items on one side and place your waste container on the opposite side. Second, do not block airflow. Air moves from the HEPA filter toward you, so avoid placing large objects in that path. Third, never place your hands over open containers. Your gloves are not sterile forever, so avoid reaching across open flasks. Fourth, move slowly. Rapid movements disrupt laminar airflow, while slow and deliberate motions help maintain sterility. And finally, avoid open flames. In mammalian cell culture, flames are generally not used inside biosafety cabinets because they disrupt airflow and create a fire risk. Contamination often comes from simple mistakes. Common sources include poor hand technique, touching sterile pipettes, reusing media bottles, overcrowded incubators, dirty water baths, talking or coughing inside the hood, shared reagents, leaving bottles open too long. Remember, you are the biggest contamination source. Let's start with bacterial contamination. Bacterial contamination usually appears very quickly. One of the first signs is rapid cloudiness in the culture media. You may also notice the media turning yellow due to a drop in pH. Floating debris may appear in the flask, and sometimes there is a noticeable strong or unpleasant smell. Because bacteria grow extremely fast, an entire culture can become contaminated within just 24 hours. Next is fungal contamination. Fungal contamination is often identified by thread-like filaments or white, fuzzy colonies appearing in the culture. Unlike bacteria, fungi usually grow more slowly. However, once established, they can spread through the culture and are often harder to eliminate. This makes early detection very important. Mycoplasma contamination is considered one of the most dangerous problems in cell culture. Unlike bacteria or fungi, mycoplasma contamination is invisible to the eye. The culture may look completely normal. There is no cloudiness and no obvious pH change in the media. However, mycoplasma can silently alter gene expression and affect cellular metabolism, which can seriously compromise experimental results. Because it cannot be detected visually, it must be tested using specific methods such as PCR, DNA staining, or commercial mycoplasma detection kits. In fact, many published experiments have later been questioned or invalidated due to undetected mycoplasma contamination. Many laboratories use antibiotics such as penicillin streptomycin in their culture media. However, antibiotics should never replace proper sterile technique. Overusing antibiotics can create several problems. They can mask low-level contamination, alter normal cell physiology, promote the development of resistant organisms, and ultimately affect experimental results. The best practice is to use antibiotics cautiously, test regularly for mycoplasma, and rely primarily on good technique rather than chemicals to maintain sterile cultures. Your carbon dioxide incubator can easily become a contamination hub if it is not maintained properly. Follow a few simple best practices. Clean the incubator regularly, ideally once a month. Replace the water tray weekly using sterile distilled water, and add antifungal agents if recommended. Avoid overcrowding the incubator, and always remove any contaminated flasks immediately. Remember, the warm and humid environment inside an incubator is ideal for microbial growth if proper care is not taken. If you detect contamination in your culture, act immediately. First, isolate the contaminated culture. Avoid opening it unnecessarily inside the hood to prevent spreading the contamination. Dispose of the culture according to your laboratory's biohazard protocols. After disposal, thoroughly clean the biosafety cabinet and, if necessary, the incubator as well. Finally, review your technique to identify where contamination may have occurred. And remember, never try to rescue a contaminated culture. In most cases, starting fresh will save far more time and effort. Developing strong laboratory habits is essential for maintaining healthy cell cultures. Always label everything clearly and record passage numbers carefully. Maintain separate aliquots of reagents and avoid sharing media between different cultures. Keep your workspace organized and maintain strict lab discipline during every experiment. In the long run, consistency is what prevents contamination. Remember, sterility is not a one-time action, it's a habit. To summarize, aseptic technique is about discipline, organization, and awareness. Your biosafety cabinet is a tool. Your habits determine success. Master sterile technique early, and your experiments will be more reproducible, more reliable, more professional. In our next Muse-B research video, we'll discuss cell passaging and maintaining healthy cultures. Thank you for joining us. If this helped you think more clearly about research gaps and hypothesis building, make sure to like, share, and comment your thoughts below. What topic should we break down next? And don't forget to follow and subscribe for more content on research, clinical science, and building strong scientific thinking.